Okay, all right. Good afternoon, everybody. So, a couple of quick housekeeping things, I guess. I mean, just again to remind you that term quiz 3 takes place Monday and Tuesday. So again, just as always, don't forget to log in at some point and complete that quiz. I will reset the Discord channels in case you're planning to use those. Other than that, I don't think I have anything else to say. Questions about things before we dive into the lecture here? Everybody's good? Nobody ever has a question? These two groups, no questions, as always. Very quiet groups. Okay. All right. Well, we, of course, were talking yesterday at the end of class about local anesthetics, which are given predominantly to basically block pain sensation. But, of course, they're going to block just nervous function in general at the local site where they are administered. And so we did a little bit about their chemistry, and I want to kind of follow up on that today because we didn't quite get it all in there. So what we had talked about yesterday was how local anesthetics all have the same chemical structure. They all have these three key parts. So they have an aromatic ring, like you see here in lidocaine. They have a terminal amine, like you see here in lidocaine. And they have some kind of linkage between those two groups. Sometimes an ester, sometimes an amide. So here you see the amide linkage in the case of lidocaine. So those are the three chemical characteristics that you're looking for to identify that something is or is not a local anesthetic. And then we started going on about how these anesthetics work. I'll just recap that quickly here. So we said that we inject these local anesthetics into the body in their protonated form. So hence, I again mentioned that it was lidocaine HCl that we were injecting here from the bottle. So it's the protonated form of the anesthetic. But that form is not membrane permeable. And so what has to happen after it's been injected into the body is that local anesthetic has to deprotonate. And then the non-protonated version, which because of the aromatic group is going to be generally membrane permeable, will then enter into the neurons. It'll reprotonate inside, and then in that protonated form, it will bind to the inside face of the voltage-gated sodium channel, blocking its ability to transmit sodium ions through it, and therefore you won't be able to get any action potentials, and so there won't be any nervous communication, there won't be any pain sensation or perception. So that's how they work. So then we started talking about how different local anesthetics have different properties. Most importantly, different anesthetics have different times to onset, how long it takes for them to start alleviating your pain, and they also have different potencies in terms of to what degree can they limit your sensation of pain. And so the time to onset, as it notes here, is related to the structure of the terminal amine, because that's where the proton is attached. And so the proton that we have to get rid of in order to become membrane permeable to come into the cells, it's that proton on the terminal amine. And so what you can see here in the figure is they're talking about how we can have different chemical groups attached to that terminal amine, and the nature of those chemical groups is actually what's going to influence the time to onset, because in order to get this proton to come off, you basically need to weaken that proton's affinity for the electrons that it's using to bind itself to that terminal amine. And so how can you weaken the affinity that proton has for those electrons? Well, if you have chemical groups added on to that terminal amine that are relatively electronegative, then they're going to be pulling electrons away from that proton towards themselves, and then that will weaken the attraction the proton has to that terminal amine, and it will very easily pop off. So again, when we think about the different local anesthetics that exist, one of the reasons that they have different times to onset is because of how they differ in terms of how easily those protons are able to pop off, which largely is related, again, to the electronegative nature of the chemical substituents that are added on to that terminal amine group. The more electronegative they are, the more they are going to pull electrons towards themselves and make it easier for the proton to dissociate, okay? So again, the more easily the proton can pop off, the faster this drug can start getting into the neurons and exhibiting its pain-relieving effects. Now, in terms of potency, the potency is largely determined by how membrane permeable is the local anesthetic. So the more membrane permeable the local anesthetic is, once it does undergo deprotonation, the more readily it can actually cross into the cell, and the more readily it can cross into the cell, the more of it can come in, and the more voltage-gated sodium channels we can inhibit. So again, they highlight here for us aromatic substitutions here on this ring. Well, you can imagine that the more hydrophobic those substitutions are, the more it's going to make the whole local anesthetic more hydrophobic and therefore membrane permeable, and therefore better able to cross the membrane to come inside the cell, and then you get more voltage-gated sodium channels that are inhibited. All right, so by contrast, if you put hydrophilic groups onto the ring, it's going to have the opposite effect, becoming less potent. Right, so again, we should be able to look at a chemical structure, identify whether or not it's a local anesthetic, and then we should be able to look at the substituents that are present on the aromatic ring and the terminal amine and get some insight into how quickly do we think this local anesthetic would act, and also how potent do we think it would be. So basically we're having this one tangential intersection of this course with organic chemistry at this point. So you kind of have to dig back into your orgochem knowledge a little bit to remind yourselves about these different substituents and their hydrophobicity, hydrophilicity, electronegativity, etc., to be able to tackle an understanding here of the chemistry of the local anesthetics. One of the things I will also just point out here, because at some point someone will ask me about it, so I might as well jump get it out of the way here now. We've said that the more easily you can deprotonate, the faster your time to onset. And then, of course, we've also acknowledged that the local anesthetic has to protonate once it's inside the cell, because it actually inhibits in the protonated form. And sometimes people will ask, well, if we've got a local anesthetic that can deprotonate very easily, so it has a fast time to onset, won't that actually make it less potent? Because once it gets into the cell, it has to pick that proton back up. The key thing to remember here is that because only the deprotonated version is coming in, you have this massive disequilibrium for that chemical reaction as that anesthetic is coming in. Right? It's coming in purely in the protonated or the deprotonated form. There's no protonated form at all, and so there will actually be quite a rapid right shift to that equation, if you will, as that deprotonated anesthetic is coming in. So the protonation inside the cell is really not a problem, because you're coming in purely in deprotonated form, creates that massive disequilibrium that is going to get rectified by just rapid protonation of the anesthetic. Yeah, that's what I said. Okay. All right, the next group of voltage-gated sodium channel inhibitors that I want to spend a little time talking about here are the neurotoxins. So neurotoxins are a bit different from local anesthetics. Obviously, they're similar in the broadest sense, in the sense that they inhibit the voltage-gated sodium channels, and therefore they can shut down nervous communication. They're different from the local anesthetics, largely in terms of where they inhibit. So this figure here, you've got this whole thing here is just one voltage-gated sodium channel, right? You can see it starts over here at its N-terminus. You can see that IFM motif along the way, and then there's the carboxy terminus on the other side. So we're looking at one voltage-gated sodium channel here, and what they're trying to show us in this figure is all the different binding sites for different types of neurotoxins. So the neurotoxins are interesting because we put them together into this one box. We call them all neurotoxins, and they all inhibit voltage-gated sodium channels. Different from the local anesthetics, though, they bind always to the external side, right? So this is the plasma membrane here, right? This would be the cytosolic side here. This is your extracellular side here. All these neurotoxins bind to locations on the external extracellular face of the voltage-gated sodium channel. which means they don't have to come in to the cell across the membrane the way the local anesthetics do. They're just binding from the outside. But what you'll also notice here is that there are 5 different sites on that external face of the voltage-gated sodium channels where these neurotoxins can bind, right? And so the diagram here very beautifully shows us different neurotoxins and the different binding sites they have. And I just kind of want to walk you through here a bit and introduce you to some of these different neurotoxins. So first here we'll start with site 1. So TTX here is tetrodotoxin, which we're going to talk more about on the next slide. We're going to focus on it in a little bit more detail. The STX there is saxitoxin, kind of very similar to tetrodotoxin. Then it talks about conotoxins, and these are kind of cool. These are a group of neurotoxins produced by animals known as cone snails. I've got a cone snail pictured there. The cone snails, basically, they have this like conotoxin-tipped radula that they basically like launch out of their body to penetrate the body wall of a prey item, and then all this conotoxin goes into the prey, and so it then paralyzes the prey by shutting down its nervous system, and then the cone snail is able to eat the paralyzed prey. So yeah, the conotoxins made by these cone snails, they're using this neurotoxin in kind of an offensive way, right? They're trying to attack something so they can paralyze it to eat it. The brevetoxins are a group of toxins that are made by dinoflagellates, which are like a type of green algae. And so, same thing, they again, basically something eats the algae and therefore consumes the neurotoxin and then gets paralyzed, well, basically, yeah, their nervous system gets shut down, they get paralyzed by it. So they're using it more in a defensive way, right, to stop things from eating them. This is also partly why when you think about like toxic algal blooms that take place sometimes in natural water bodies, this is part of what is concerning about these things, because there's all these toxins like neurotoxins that these algae are producing, and so if they suddenly are increasing in a water body in tremendous numbers, then it becomes unsafe for people to be out there where they could potentially be, swallowing water containing neurotoxin-containing algae. The cingulatoxins, I don't have a picture to go with this. They're a toxin that is made by some groups of fishes. They bind, as you can see here, to the exact same site as the brevetoxins do. Then site 4 talks about scorpion and spider beta toxins. So basically, again, scorpions, as you probably know, they have their little stinger tail that they can use to inject neurotoxins into their prey or even to defend against a predator. Spiders the same way. They have neurotoxins that they can use to inject with their fangs in order to, again, paralyze prey or perhaps to even defend themselves against possible predators. The batrachotoxins that are mentioned there, site 2 binders, these are actually the neurotoxins that you find in the skin of poisonous frogs, right? So these very bright colored frogs that are warning you that, hey, I'm full of toxins, don't come here and eat me. So they make these batrachotoxins that are neurotoxins that can bind and inhibit voltage-gated sodium channels. The grayanotoxins that they mentioned there, which bind to the same site, these are made by a number of different flowering plants. One of the best well-known of these is rhododendrons, which are pictured here. So again, they're basically a deterrent against herbivory. And then lastly, again, scorpion and sea anemone toxins. So we've already talked about scorpions, but sea anemones, which are cnidarians, they again are very similar to the conotoxins. They actually have stinging cells on their tentacles that they can use to inject these neurotoxins into the body of their prey. So yeah, a cool group of neurotoxins here. A cool group of chemicals that can shut down your nervous system by inhibiting your voltage-gated sodium channels. My comment here that I have on the screen is that neurotoxins are usually much more potent than local anesthetics. So they can actually bind to the voltage-gated sodium channels, even at relatively low concentrations. So they're very potent inhibitors. So, nature has done a great job of creating these really quite toxic poisons. Next up here is I just want to go into a little bit more detail about tetrodotoxin. So you can see the chemical structure of tetrodotoxin here. And again, it just shows us, as I already noted, that it basically binds to the extracellular side of the channel and inhibits from that direction, and in doing so prevents sodium from moving through, so we can't generate action potentials. Where does tetrodotoxin come from? Well, there are a variety of species that make tetrodotoxin. One of the most well-known is the pufferfish, right? So this pufferfish here has a couple of tricks up its sleeve to try and deter predators from consuming it. One is what it's doing right now, where it has puffed itself up to this very large size. So these guys will basically, puff themselves up, hence the name pufferfish, to try and make themselves look bigger than they actually are. And obviously they got all these little spines on their surface, so I mean they're supposed to look like a spiky ball that you wouldn't want to mess with. But in addition to that, in their liver and in their ovaries in particular, they also synthesize tetrodotoxin. And so they have this chemical deterrent against being eaten as well. I could remember the very first time that I ever learned about pufferfish was actually courtesy of The Simpsons. There's a Simpsons episode where this kind of unskilled Japanese chef is trying to prepare pufferfish for Homer. It's a delicacy because you have to very skillfully prepare it, because if you don't and you end up contaminating the dish with the tetrodotoxin that is in the fish, then ultimately, of course, the person dies. So you can imagine, if you were eating something, I mean, maybe you think this every time you go to a UTSC food place and you think, am I going to die from eating this today? But this is actually a legitimate thing to be concerned about, I guess, if you're eating pufferfish, that if it's not been prepared correctly and expertly, the contamination of tetrodotoxin from the various organs that produce it could make that your last meal. Another place where, again, there's a lot of pop culture surrounding pufferfish. This is another show that I really like, one of my favorite television shows of all time. I don't know if anyone's familiar with the detective show Columbo. Anybody ever heard of this show? It's from the 1970s, so I have all the episodes on DVD if anyone wants to borrow them at some point. There was an episode, so it's a typical kind of detective show, and they had this one episode in one of their seasons where they had this really hoity-toity chef who was being blackmailed by some people, basically murdered them by extracting tetrodotoxin from the pufferfish and injecting it into their wine, and thought he was going to be clever and get away with it because who's going to know about this? And of course, as you can imagine, he gets caught by this really intelligent detective who outwits him. So anyways, pufferfish, big things in pop culture. One of the things that might be going through your mind right now, especially going back to the pufferfish and just eating it, is like, well, why don't we just cook this thing? Can't you just cook the pufferfish and have the heat break down the tetrodotoxin? The answer is no. Tetrodotoxin is actually remarkably heat stable, so it's pretty difficult to just cook it away. So that's a point to be mentioned here. But the other thing I wanted to also mention is that there are actually some animals that have figured out how to circumvent the tetrodotoxin toxicity, at least to some extent. And one such species is this species of snake right here. So there was a study that was done a few years ago. They were looking at two different snake species, so this T. sertalis and this T. ordinoides. And you can see that for T. sertalis, there are two different populations, one from Idaho, one from Oregon. It turns out that the T. sertalis snakes that live in Oregon, they basically eat newts that make tetrodotoxin. And so obviously when biologists discovered that there were these snakes eating these tetrodotoxin-laced newts, they were like, well, how are they able to do this, right? Why aren't they dying as a result? And so first and foremost, just to show you that they aren't dying, or at least not being affected as much, this research group basically gave these different snakes, so the T. sertalis snakes in Oregon, and also the ones in Idaho, which don't eat the toxic newts, and then this other species in Oregon, which also doesn't eat the toxic newts, this T. ordinoides, they gave them different levels of tetrodotoxin, so they basically just injected it into them, and then they measured how much it was affecting them by measuring how fast they could actually slither around. And you can see that the T. ordinoides and the T. sertalis from Idaho, the ones that don't eat toxic newts naturally, they start to pretty rapidly, lose their ability to move at relatively low concentrations of tetrodotoxin. But you can see here that the T sertalis, it requires quite a bit higher tetrodotoxin level before it starts to show any effects. And you might look at it and say, like, this doesn't look like a very big shift, but keep in mind, this is a logarithmic scale down here. So, I mean, that shift right there is kind of on the order of at least a 10-fold difference. Right? So ultimately, those T sertalis in Oregon there, the ones that are eating the toxic newts in their natural environment, they're like 10-fold more resistant to the toxic effects of that tetrodotoxin. So the point here is it's possible for animals to evolve a tolerance that are resistant to tetrodotoxin, and the way they do that is largely by making little changes to the chemical structure of their voltage-gated sodium channels, right, to make it such that the tetrodotoxin doesn't bind to the voltage-gated sodium channels quite as well, and then it doesn't inhibit quite as well, and therefore you can be more resistant to it. Okay, the next thing I want to talk about with respect to action potentials is something called the all or none principle. And this is the basic idea that action potentials either happen or they don't, right? So either we reach the threshold stimulus and we get an action potential, or we don't reach the threshold potential and we don't get an action potential, and that's what they're trying to illustrate here. This is the all or none principle. Right, so on the bottom graph, they're showing us the stimulus strength, and on the upper plot here, the membrane potential that results. And so you can see here that for the this stimulus here, it's described as a subthreshold stimulus because even though it caused a little bit of a depolarization, you can see it did not get us anywhere near the threshold potential, so no action potential occurred in response. The second stimulus here is a little bit stronger, but again, still a subthreshold stimulus, does not get us anywhere near the threshold, so no action potential occurs. The third stimulus is described as a threshold stimulus because it was just strong enough to get us to the threshold potential, and because we got to threshold potential, an action potential occurred. And then the fourth stimulus here is called a supra-threshold stimulus because it's stronger than the threshold stimulus. But again, what we have to appreciate is because we got to threshold, we still just got an action potential as a result. So whether we had a threshold or a supra-threshold stimulus, the end result here was the same. An action potential occurred, right? And when we had sub-threshold stimuli, we didn't get an action potential. So again, that's the all or none principle, right? You don't have bigger action potentials in response to supra-threshold stimuli. Anytime you get to the threshold or above, you get an action potential. And it's the same action potential no matter how strong that stimulus is, right? We all know the all or none principle. I mean, think about flushing a toilet, right? And I'm just talking about a regular toilet, right? Not fancy toilets, just regular toilet with a flush handle on it, okay? Because people keep imagining like, well, what about this toilet and this toilet and all these fancy toilets? Flush toilet with a little handle on the side. Right, when you go and you've had your massive, you've pooped in the toilet and you go to flush, right, you push on the handle a little bit, it doesn't flush, push a little harder, it doesn't flush, eventually you get to the point where you cross the threshold and it does flush, right? But if you did go in there and like really drop a big one, you can't go in there and just like jam the handle down and think, oh, that'll flush it real hard and maybe you'll actually get it down there. It doesn't work that way, right? It's an all or none principle. Right? It either flushes or it doesn't. Doesn't matter how hard or light you press on that handle. All right? That's an important life lesson. Right? When you have kids at some point, you're going to remember this day where you're like, hey, kids, right, just because you took a big poop today doesn't mean you have to jam on this handle and break the toilet. Right? Mommy and daddy aren't made of money. We can't buy toilets all the time. So just keep that in mind. You can teach them about the all or none principle right there when they're potty training. Now, of course, the question that comes up when you are thinking about this all or none principle is, well, are we not losing information here then? Because if we have a threshold stimulus and we get an action potential, and then we have a super threshold stimulus and we get the same damn action potential, like when we're sending that action potential to other parts of the nervous system, don't we lose the information about how strong the original stimulus was that caused this action potential to occur? Isn't that useful information that the nervous system wants to know? So how do we capture that information? The answer is we don't do it through individual changes to a single action potential. Rather, that stimulus information is coded in the frequency with which action potentials are being sent. And so I'm going to introduce you at this point to something called strength frequency coding. And so we have a very similar plot to the last one here. We've got our stimuli being shown there on the bottom plot, right, in the same kind of way that we have seen them in before, where you just kind of have these little kind of ups to show a stimulus is being applied, and the more it goes up, the stronger the stimulus is. And then at the top, we have our membrane potential being measured. And so in the first case here, we have a slightly subthreshold stimulus, as you can see. And so there would have been a gradient potential that would have occurred. They're not showing the gradient potentials here, but there would be a little gradient potential, but it wouldn't get us to the threshold, so no action potentials occur. Here in the middle, we have a slightly suprathreshold stimulus. So you can see now that action potentials are occurring, and they continue to occur the entire time that that stimulus is in place. Right? We keep generating action potential after action potential as long as that stimulus is in place. But notice the frequency of the action potentials here. We had 4 action potentials generated in, say, that 1 millisecond stimulus time. Right? That's our frequency. But then in the third case here, we have a really suprathreshold stimulus, a much stronger stimulus. So obviously we have action potentials occurring, again, throughout the entire time the stimulus is in place. But now notice how the frequency of those action potentials is much greater, right? We've generated maybe 10 action potentials in that same millisecond. So we code the stimulus strength not in changes to an individual action potential, rather we code that stimulus information in the form of action potential frequency, okay? Now, one point to, I guess, mention about that is that we talked the other day about the absolute refractory period, right? This period of time during which you absolutely cannot generate another action potential. And as it says here, obviously the length of the absolute refractory period is going to determine the maximal frequency at which we can generate action potentials, right? So that's one thing to keep in mind. There is an upper limit to how many action potentials we can generate per unit time. And so at some point, even this strength frequency coding is going to kind of tap out, right? There's going to be kind of an upper limit where you're just like, hey, we can keep going to stronger and stronger stimuli, but we can't actually generate action potentials any more frequently because the limiting factor now becomes the length of the absolute refractory period. The next thing I want to talk about here is, well, what's the underlying mechanism causing this strength frequency coding? Like, why does a stronger stimulus, or how does a stronger stimulus actually lead to a higher frequency of action potentials? And so I have this figure here, which is going to help us to talk about that. And I'm mostly going to direct your attention to the very bottom of this figure, where it's talking about the excitability of a neuron as a function of time. So excitability is a measure of, like, how easily can we get this neuron to generate an action potential? Like, how responsive is this neuron? How capable is it of generating an action potential? And so you can see here that when we are at resting membrane potential, right, or even if we're, just below the threshold in general, the excitability of a neuron is very high, right? So that's because we've got all of our voltage-gated sodium channels in their closed state, and we know from our conversation yesterday we can open up those channels from their closed state and therefore generate action potentials. So when we are below the threshold potential, in this case before any action potentials have occurred, we have this very high excitability. It's very easy to stimulate this neuron to generate an action potential. We can see that once we actually generate the action potential and we're above threshold, we're now in the absolute refractory period. And as we discussed yesterday, the excitability during the absolute refractory period is 0. Right? We cannot whatsoever generate an action potential during the absolute refractory period because either the voltage-gated sodium channel is already open and we cannot open it further, or it goes into the inactive state and we cannot open from the inactive state. Now here's the key to this whole strength frequency coding thing. It's that once we come back down below threshold and we now go into the relative refractory period, we start to see this gradual increase in excitability over time. So excitability returns It goes above 0 because once you go below the threshold, the voltage-gated sodium channels go back to their closed configuration, and we can open them from that state, so we can generate another action potential now. But remember, as we discussed yesterday, the voltage-gated potassium channels are only slowly closing during this period of time. So because there's still lots of them open, right, especially right at the very beginning of this relative refractory period, you got lots of voltage-gated potassium channels that are still open. There's, lots of potassium ions crossing the membrane, trying to hyperpolarize that membrane and oppose the ability to get back up to that threshold potential. That's why initially, although the excitability is not 0, it's still very low. But as more and more of those voltage-gated potassium channels close over time, right, that hyperpolarizing effect of potassium becomes gradually lesser and lesser, and so the excitability is gradually increasing. And then eventually we get to the point where all the voltage-gated potassium channels close, we're back to resting condition, we're back to our high excitability. Now the key here is if you have a very strong stimulus, right? If we had a very strong stimulus that evoked this first action potential, and we keep that very strong stimulus in place, as soon as we get into the relative refractory period, that really strong stimulus, even though the cell is only minimally excitable at that point, that strong stimulus will be enough to generate another action potential. So we'll get two very closely spaced action potentials. And if we keep doing that over and over, we're going to have a lot of action potential frequency. But imagine instead that we had a weak stimulus. So a weak stimulus caused that first action potential to occur, then we keep that weak stimulus in place. That weak stimulus is not able to actually cause another action potential to occur until the excitability gets a little bit higher, and then it's able to cause that next action potential. But now those two action potentials are spaced out in time a lot more, so their frequency is a lot less. So this is the reason why we have this stimulus strength, action potential frequency relationship, right? So the slow closing of the voltage-gated potassium channels is actually key to this whole thing, right? Because it means that the excitability of the neuron only gradually increases over time after the first action potential, and that allows, therefore, for strong stimuli to be able to cause more frequent action potentials, because it can deal with that low excitability that happens right at the beginning of the relative refractory period. But weak stimuli are going to lead to lower frequency action potentials because they can't generate that next action potential until a good number of those voltage-gated potassium channels have closed and you return the excitability to a higher value. All right, the next thing to talk about here, I think it's actually the last thing we have to talk about in this electrophysiology of neurons lecture, is to talk about the propagation of action potentials. So we know that action potentials are going to be generated initially at the axon initial segment. If we get to threshold, it gets generated. But then we have to propagate those action potentials all the way down the length of the axon and into the axon terminal branches, all the way to their terminals, so we can trigger the release of neurotransmitters, which of course is going to be the next topic that we'll start into sometime next week. So first of all, the word propagation literally means, or literally what the term means is conduction without decrement. So it's this idea that action potentials can be conducted down the entire length of the axon without decaying, meaning that the action potential that you generate at the axon initial segment, goes up to like positive 30. All the action potentials you generate down the entire length of the axon, they're all going to be equally as strong, right? And again, this is a direct contrast to graded potentials, which we know undergo decremental conduction, which is what makes them a very localized phenomenon. Again, the fact that you can propagate action potentials without decrement is what allows them to travel long distances. So again, that contrasts with graded potentials. So let's talk about how action potentials propagate. And so we've got this figure here to help guide this conversation. And so what you're looking at here is an axon, as it says. So this would be the cytosol of the axon through here, right? The black lines would represent the plasma membrane, and so then the white space out here and out here would be the extracellular fluid. All those little kind of black, they kind of look like H's maybe, these little black things here, embedded in the membrane, these are voltage-gated sodium channels. So what you can see here is in this area that's kind of highlighted in purple, you have voltage-gated sodium channels that it says are open in that region. And so then, not surprisingly, that region is currently in the depolarization phase of an action potential. Because whenever the voltage-gated sodium channels are open, you've got sodium coming into the cytosol of the neuron and making the membrane potential more positive, so we're depolarizing phase. To the right, so downstream, you can see that all the voltage-gated sodium channels there are currently closed, right? Because there's no stimulus in that location to trip them open. But what happens here is as sodiums come in through the open voltage-gated sodium channel, as we've talked about before, they can perceive that this area over here is currently at its resting potential and that there are negative charges in the cytosol over there. So just like we had seen before, these sodiums come in through the open voltage-gated sodium channels. They perceive, oh, down here there are negative charges, let's start going down there. And so that's what they're trying to show us with those arrows. The sodiums start going down here, attracted to those negative charges in the cytosol, and as they shift down there, they depolarize that region up to the threshold, and then the voltage-gated sodium channels in that area respond by opening. And now this region of the axon, you can see this is now 1 millisecond later, right, compared to where we started, because those sodiums moved downstream and depolarized this region and got it to the threshold, now these voltage-gated sodium channels in that region are tripped open, and an action potential begins to occur there. And then sodiums come in through those newly opened channels, and they migrate downstream, attracted by the negative charges, and then they depolarize that region to threshold, and then those voltage-gated sodium channels open, and that just keeps happening over and over and over again. This is how we can propagate the action potential down the length of the axon. Now, the other thing to think about here is, well, when the sodiums come in, you know, they show some of them going backwards here to this region over here in green that has already experienced an action potential. Right? The action potential in this case is starting over here and traveling in this direction. So this region over here just underwent an action potential. But some of these sodiums that are coming in through this open voltage-gated sodium channel, it shows them migrating backwards. How come they don't trigger an action potential to occur back upstream? How come we only get action potentials happening downstream where they haven't already occurred? Well, there's a couple of explanations here. One is they already highlight for us that this region, having just gone through an action potential, is likely going to be in the refractory period, right? So either the channels there will be, you know, inactive currently and can't be opened at all, no matter how many damn sodiums come back there, or even if we're in the relative refractory period, as we just established, it's harder to open those channels because you've got this hyperpolarization that's happening that's opposing any of the backflow of those sodiums. Okay, so that's one reason, right? The other reason would be that if this region is still, you know, not down below resting potential yet, let's say it's maybe in the absolute refractory period, so we're still above the threshold, that would also mean that this region would not be as negatively charged on the inside as this region here. So the sodiums would be more strongly attracted to go downstream where they sense more negative charges rather than going back upstream where the charges might still be more positive. So it's kind of some combination of those things, the totality of those things, that's ultimately going to ensure that action potentials propagate unidirectionally, right? They propagate from the axon initial segment down to the axon terminal, and they don't try to back propagate, you know, turn around and go backwards at some point or start going out in both directions. It's because of the effect of the refractory period predominantly, coupled with the possibility that if you're still at a more positive resting or a more positive potential, because maybe you're still above threshold or something, that those sodiums would just not be as attracted to that lesser negatively charged region than they would be to the region downstream that's still at resting potential and therefore very negative, and they go that way instead. Okay, the next thing for us to think about here with respect to propagation of action potentials, now that we kind of understand how they get propagated unidirectionally from the axon initial segment down to the axon terminals, is to think a little bit about the speed at which action potentials can be propagated. And this is important because the faster that action potentials are propagated, the faster that we can communicate nervous information through the body. Right? Obviously, if I'm standing here and, suddenly my hand drops this, my clicker, and it starts to fall, and I want to grab it before it hits the ground, that fast reaction time requires an underlying fast action potential propagation to allow me to immediately sense that I've let go of it, to immediately contract muscles so I can grab it before it hits the ground and smashes into bits and pieces. So having fast action potential propagation speed can be really important in a lot of circumstances. So before we get into the nitty-gritty details here, I just want to use this little figure here to kind of illustrate what I think is a very kind of basic principle that we have to wrap our head around here. So as they're kind of showing in this figure, right, so here is our axon initial segment where the action potential is first generated. And if our goal is to propagate that action potential to the axon terminals here as quickly as possible, what we want is for the sodiums that come in through the voltage-gated channels here, we want those sodiums to come in, and then we just want them to travel down the axon within the cytoplasm as far as they can go, right? The more time that the sodiums spend traveling down the axon within the cytoplasm, the faster we're going to be able to propagate action potentials. What slows action potential propagation down is when we have those sodium ions, for example, coming in, traveling a bit, then leaking back out, and then we have to maybe open up another voltage-gated sodium channel to get more sodiums to come in. The more time that ions are spending going this way, perpendicular to the direction of their travel, leaking across the membrane and then having to bring them back in through other voltage-gated sodium channels, the more time they spend going in that direction across the membrane, the longer it's going to take to get that action potential from the initial segment down to the hillock, or down to the terminal, right? It's kind of, if you imagine that, I said, okay, I want you to leave this campus tonight and get home as fast as possible. Maybe your mom says, hey, I want you as fast as home. I want you home as fast as possible after lecture today, right? We've got things we got to do. I need you home. The more that you can get on the highway and just drive straight down from here to your house, the faster you're going to get there. The more that you're driving along and you're like, oh, an IKEA, and here I go. Oh, there's a Sephora, here I go, right? The more time you spend traveling in directions opposite of the direction you're trying to go, the slower it's going to be. So our goal is the same with action potential propagation. We want sodiums to come in through the voltage-gated channels and spend their time traveling down the cytosol to get to the axon terminals. The more time they spend leaking out, the more time we have to regenerate new action potentials coming in through voltage-gated channels, the slower the process is going to be. So with that in mind, we can now think about what's called the length constant, which is kind of the way I like to think about action potential propagation speed. So the length constant, which is symbolized here by the Greek letter lambda, the way to think about the length constant is it's basically the distance that a sodium ion can travel. I shouldn't say it that way. It's the distance that a graded potential can travel before it decays to about 37% of its maximum value. Okay, so that's the formal definition. But, practically speaking, what we're saying here is when you have a depolarization that takes place, right? In this case, it could be a massive depolarization that takes place as part of an action potential, right? When those ions come in, the further they can travel down the axon in the cytoplasm before they leak out and kind of bring the membrane potential back to rest, the longer that length constant is, that distance they can travel before they leak out and kind of decay that depolarization back to rest, right, the more time that we are spending traveling down the cytoplasm, the less time we're spending leaking out, right? So the length constant is proportional to action potential propagation speed, because the length constant is a measure of how far did this ion go before it detoured off to the Ikeas and the Sephoras and we had to bring new ions in, right? So the longer the length constant, the more that those ions are just traveling down the length of the axon, not leaking out across the membrane, where then we have to bring new ions in, and so that should lead to faster action potential propagation. Now, you can see here that the equation tells us that there are two factors that determine what the value of the length constant is. One of these factors is the resistance of the neuron membrane. So that's how easily can ions leak out across the plasma membrane, right, to get back out of the cytosol, to get back into the extracellular fluid, which of course brings us back to the resting potential. And obviously if ions are leaking out across the membrane, right, then they're not traveling in the desired direction. So we want the membrane to be as resistant as possible to the leak of ions across it. We want sodiums that come in through open voltage-gated channels to not be able to leak back out, right, because then they're moving in the wrong direction. We want these voltage-gated sodium channels to open, let sodiums come in, and we want the membrane to be so resistant to the passage of ions across it that those ions come in and they're like, okay, well, I got no choice. I'm going to just keep traveling down the cytosol here, right? There is no way for me to leak across that membrane. The membrane is so resistant to my passage. Going back to my get home quickly analogy, it's almost as if you're like, okay, well, if there are no off-ramps that allow me to get off the highway to get to the Sephoras and the Ikeas, then I have no choice but to keep driving towards mom. So I get there faster. But by contrast, if there's off-ramps everywhere leading to all these fun places you want to visit, then you're driving along, you're getting off, coming back on, oh, get off over here, right? There's no resistance to you getting off the road, so you constantly do, and then it slows down the whole journey. So that's the idea here. The membrane needs to be as resistant as possible to the flow of ions across it, so that these ions, once they come in through open voltage-gated sodium channels, have no option but to keep traveling down the cytosol. And then the other part of the equation here is the internal neuronal resistance. It's a measure of how hard is it for ions to travel down the cytosol here, travel within the neuron. We want that to be as low as possible because it's in the denominator. And that makes sense too, right? If these sodium ions come in through this open voltage-gated sodium channel, and then they're like, oh, it's really cramped and hard to move down the cytosol here, they'll just leave. They'll go back across the membrane then. Again, go back to my car analogy. It's like you driving, right? And you're driving along and you're like, oh, there's traffic jams. I can't get anywhere. Oh, Sephora, I'll just go here, right? So our goal is to make it hard for ions to leak out and easy for ions to flow within the cytoplasm. If we can do those two things, then ions come in through the open voltage-gated channels. They're most likely going to travel all the way down the cytosol to get to those terminals as quickly as possible. We'll have fast action potential propagation speeds. Does that make sense? Okay, so that's the general idea. The specifics will have to wait until Tuesday, so you'll have to spend your weekend just waiting to learn about specific mechanisms of action potential propagation. I'm sorry if that ruins your weekend, but we'll all be okay, I think. I will see you on Tuesday. Don't forget the quiz, Monday, Tuesday, and I'll see you Tuesday.